Up to 35% of BAS people have Rh negative blood. And Rh negative blood is one of the most unusual.
A 25-year-old woman nearly dies from A, blood transfusion from her own husband. Same blood type, same match on every test. But in 1939, medicine finds that her body tries to destroy it anyway. She is 33 weeks, pregnant when she is admitted, and the pregnancy has already gone wrong. The baby she delivers is stillborn, 1 pound 5 ounces, small, enough to fit in 2 hands. She starts bleeding badly. The obstetricians do what obstetricians did in 1939 for A, hemorrhage. They give her blood from the most obvious donor in the building, her husband, and because his blood type is compatible with hers on paper, nobody expects trouble. What happens next has no explanation on any chart in that hospital. She reacts violently. Pain, fever, urine turned the wrong color. 2 doctors, Philip Levine and Rufus Stetson, test her blood against sample after sample from the hospital's donor pool. Her serum attacks 80% of them. Blood, that by every rule of the system is supposed to be safe for her. Levine and Stetson published their case in JAMA on July 8, 1939. And their conclusion is the strange part. Whatever reacts as violently in her blood was not there before her pregnancy. It had to be inherited, and the only person who could have given it to her is the baby she just lost. Read that again. The child that just died left something behind in its mother's bloodstream, something her immune system had learned to fight like a foreign invader. Not an infection, not a disease she caught. Her own son or daughter. That single case was the first recorded sign of a mechanism that at the time had no name. A mother's own immune system learning to attack her unborn children quietly, killing infants year after year with nobody able to say why. It would go on for 3 more decades before anyone found a way to prevent it. And to understand how a woman's body ends up at war with her own child, you have to start with what is actually different about her blood, which is not what almost anyone has been told, right down to the name printed on the chart. Here's the correction almost nobody watching has ever heard, even people who know. Chapter 1, a gene that isn't there, their own blood type, well, Rh negative, is not a mutation. It's not a variant. A glitch or a weaker version of Rh positive, sitting 1 step lower on some genetic ladder. In most people of European descent, it isn't a change to a gene at all. It's the absence of one. The RhD gene, the gene that makes the D antigen on the surface of a red blood cell, is simply not there. Deleted, gone from the chromosome, like a page torn cleanly from a book rather than a sentence, crossed out on it. And it gets stranger because the name on the chart is wrong too. The Rh in your blood type is not named after anything human. In 1940, Carl Landsteiner and Alexander Wiener injected rabbits and guinea pigs with blood from a rhesus macaque, and the antiserum those animals produced reacted with about 85% of human blood samples, close enough to the frequency Levine's human antibody had already suggested that everyone assumed the two were the same thing. They weren't. Differences showed up as early as 1942, and by 1963, the case was clear. The monkey antigen was a completely different molecule, and it was renamed LW after Landsteiner and Wiener themselves. But Rh was already in universal use for the human antigen, printed in every textbook and on every hospital chart, and nobody went back and fixed it. Every time you have seen Rh negative on a form, you have been reading the ghost of a naming mistake medicine noticed, shrugged at, and kept. If a naming error this basic has survived in medicine for more than 80 years, and this is the first time you're hearing it, subscribe and follow the mysteries history tried to Very. So how? Does an entire gene go missing? It isn't. Mysterious once you see it. RHD sits, sandwiched between two almost identical stretches of DNA, roughly 9,100 base, pairs each, called rhesus boxes. Think of them as two copies of the same, bookmarks sitting in the same book a few 100 pages apart. When a cell copies that stretch of chromosome, those nearly identical bookmarks can fool the copying machinery into lining one up against the other instead of against itself, and everything between them falls out. The gene gets cut away, and what is left behind is a single hybrid bookmark where two used to be, half of one and half of the other. That hybrid is a scar geneticists can still read today as the fingerprint of exactly what went wrong. Wagner and Flegel described it clearly in the journal Blood in 2000. RHD gene deletion happened in the rhesus box. That deletion is the main European path to Rh-negative blood. Other populations reach the same result by different genetic paths, but this is the one behind most Rh-negative people watching this video. And here's what surprises people who think they understand their own blood type. Rh isn't one switch. It's a whole system of antigens. And D is just the one that matters most, clinically, because it's the one the immune system reacts to most strongly. When you're told you're positive or O negative, that sign is entirely about whether you carry that one antigen. Roughly 15% of people of European descent don't. None of this, the deletion, the system, the borrowed monkey name, is dangerous on its own, and absence is just an absence. It becomes something else entirely the moment a woman carrying and conceives a child who doesn't. Picture one family instead of a statistic, because that's the only way this makes any sense. A woman is Rh negative. Her husband is Rh positive. Their first child inherits his father's. Chapter 2, the second child, D antigen, Rh positive, like most of the population, and the pregnancy is completely uneventful. She delivers a healthy baby and goes home. And by every measure available to her doctor, nothing at all went wrong. Here's why. For almost a whole pregnancy, a mother's blood and her fetus's blood never actually mix. They move in separate, closed circulatory systems, and the placenta keeps them apart. Nutrients and oxygen cross, but the blood cells mostly don't. The one moment that changes is delivery, when the placenta detaches and a small amount of the baby's blood slips into the mother's bloodstream. For a first pregnancy, that exposure comes right at the end, with almost no pregnancy left for it to matter in. But her immune system does not forget what it just met. It has now seen an antigen D that her own body has never carried, and it starts building a response the way it would to any foreign invader. That first response is slow, built from antibodies too large to cross the placenta, even if she got pregnant again right away, which is exactly why the danger is invisible the first time. It's the second Rh positive pregnancy where the story turns. Her immune system already has a memory of that antigen, and this time the antibodies it produces are smaller, small enough to cross the placenta directly into the fetus's bloodstream. While it's still inside her, while it matters most, those antibodies attack the fetus's Rh positive red blood cells as foreign, breaking them down faster than the fetus can replace them, and everything that follows comes from that one fact. We know all of this because of the same man who started this story. 2 years after that 1939 case, Philip Levine went back and did the work properly with Lyman, Burnham, Eugene Katzen, and Peter Vogel. He published a paper in the American Journal of Obstetrics and Gynecology in 1941, volume 42, pages 925 to 937, titled The Role of Isoimmunization in The pathogenesis of erythroblastosis. Fetalis. That title is doing something huge quietly. It says this: disease is not a mystery of pregnancy or bad luck or something wrong with the baby. It is an immune reaction in the mother against the Rh factor on her own child's red blood cells, and in the cases Levine and his colleagues studied, that mechanism explained 93% of them. One paper, and the most common, unexplained way to lose a newborn had a cause. The numbers under that cause are worth holding on to because they explain why the disease was everywhere and invisible at the same time. Roughly 15% of women of European descent are Rh negative, and the great majority of their partners are not. So the setup for this disease sits inside a large share of ordinary pregnancies. Without prevention, about 16% of compatible Rh negative mothers carrying an Rh positive baby went on to develop the anti-D antibody at all, and in a second pregnancy with another Rh positive baby, roughly 6% of Rh negative women ended up with a child actually affected by it. Small percentages applied to nearly every family in a country add up to a body count. What affected means in practice: anemia, because red blood cells are destroyed faster than a fetus can make new ones; severe jaundice after birth, because that breakdown floods a newborn system with bilirubin faster than an immature liver can clear it. In the worst cases, kernicterus, permanent brain damage from bilirubin crossing into brain tissue. In the most severe cases, hydrops fetalis total, body swelling from heart failure, the heart struggling to make up for blood it no longer has enough of, and worst of all, stillbirth, where the script started. Here is the cruelty inside this disease almost nobody names plainly. The danger isn't caused by a sick pregnancy. It's caused by a healthy one, by the first baby that came out fine. Every pregnancy that followed was more dangerous than the one before it, and families had no way of knowing why. A mother would watch her children get sicker by birth order, sometimes losing one completely, with nothing in the medical understanding of the day to explain why. If your family has a story like that buried in it, a baby who didn't survive, a grandmother told quietly to stop having more children and nobody ever explained why, I want to hear it in the comments, because this is very likely the reason nobody could. For 30 more years after Levine and Stetson's discovery, medicine could not stop this from happening. What it could do, starting in the 1930s, is amazing given how little anyone understood the cause. Between Levine and Stetson's 1939 case and 1968, medicine did not stand still. It just couldn't reach the actual cause. So it did the next best thing. It tried to survive the disease after it had already started. The first breakthrough wasn't even a treatment. It was a Chapter 3, before anyone could stop it, diagnosis. In 1932, physicians Lewis, Diamond, and Kenneth Blackfan noticed something nobody had connected before: stillbirths with terrible total body swelling, severe newborn jaundice, and unexplained newborn anemia were not 3 separate problems. They were 3 faces of the same disease. Diamond and Blackfan named it erythroblastosis. Fetalis, from the immature red blood cells, erythroblasts, that flooded into these infants' bloodstreams as their bodies tried desperately to replace what was being destroyed. What they still didn't have was a cause. They had correctly identified the single deadliest hemolytic disease in newborn medicine. 9 full years before Levine's 1941 paper told anyone what was causing it: a disease with a name, a description, and no explanation at all. The first real treatment came in October 1946, and it is a startling piece of medicine for its time. Lewis Diamond, working with Fred Allen, performed what is called an exchange transfusion threading a catheter into a newborn's umbilical vein and in careful cycles, removing the infant's own damaged blood and replacing it with healthy blood, basically swapping out the baby's entire blood volume after birth. It sounds almost too aggressive to be real. It worked. Mortality from erythroblastosis. Fetales fell more than tenfold. Once exchange transfusion became standard practice, and Diamond published the results in Pediatrics in 1948. But exchange transfusion could only save a baby after birth, and plenty of the worst cases never made it that far. The disease was killing fetuses in the womb weeks before delivery, and no catheter reaches a baby who hasn't been born yet. That stayed unsolved until 1963, when New Zealand physician William Lilly did something that on paper sounds reckless. He transfused blood directly into a living fetus, still inside its mother, through a needle passed through her abdominal wall. Lilly didn't set out to invent this. He stumbled into it during a routine amniocentesis. A needle was accidentally placed into a fetus's abdominal cavity instead of the surrounding amniotic fluid, and that accident showed him the fetal abdomen could be reached safely, enough to try something on purpose. His first 3 attempts at an actual intrauterine transfusion failed. The 4th was performed on a fetus, expected to die before birth, and it survived. Lilly's early technique carried a success rate of only around 40% worse than a coin flip, and yet it was the first time in the history of this disease that a doctor could reach into the womb itself and change the outcome before delivery. Notice what every one of these breakthroughs has in common. Diamond's exchange transfusion, Lilly's intrauterine needle. Each is a rescue attempt arriving after a mother's immune system has already decided to attack her child. Nothing in 1932, 1946, or 1963 could stop that immune system from learning to see its own baby as the enemy. Doctors had gotten good at treating a fire already burning. Nobody had found a way to keep it from starting. The answer, when it finally arrived in 1968, wasn't a better rescue. It was something almost unbelievably simple, and almost nobody expected it to work. Here is the idea that makes 1968 the peak of this whole story, and it is truly strange when you say it out loud. To stop a mother's immune system from. Chapter 4, 1968. Learning to attack her baby. You give her the very antibody her body would eventually make on its own. Sit with that because it sounds backward. The problem is antibodies attacking Rh, positive blood cells, and the fix is injecting more of that exact antibody into the mother. Here's the logic. Under it, elegant once you see it, the injected antibody clears the small number of fetal Rh positive cells out of the mother's bloodstream before her own immune system gets a proper look at them, before her body has the raw material to build its own long-term memory response. You're not fighting the antibody. You're using it as a decoy, handing the immune system an answer so complete it never has to work out the question for itself. The people who turned that idea into medicine were Vincent Frieda, an obstetrician at Columbia, John Gorman, a pathologist also at Columbia, and William Pollack, a research scientist at Ortho Pharmaceutical. Between them, they had the clinical need, the immunology, and the industrial capacity to make it scale. And to prove it worked, they needed volunteers, willing to be deliberately exposed to a disease that until then had only ever been treated as an emergency. Here's the honest part most retelling skip. Their early trials used Rh-negative male volunteers, and some of those volunteers were prisoners at Sing Sing. They were injected with anti-D antibody and then deliberately with R-positive blood cells, the same exposure that would put a pregnant woman's fetus at risk, engineered on purpose in men who could not have a pregnancy at all. The result was as close to perfect as A. Trial gets. None of the men who received the antibody first went on to develop their own immune response against D. 8 of the 13 untreated controls did. The mechanism worked exactly as predicted in humans, and there was no more reason to wait. The FDA approved it in 1968. The UK approved it the same year. And what happened to the death toll over the next 7 years is one of the fastest reversals of a mass killer in the history of modern medicine. In 1968, Rh hemolytic disease killed 941 infants in the United States, 2.7 for every 10,000 live births. By 1975, that number was 269, 0.9 per 10,000, a 71% drop in 7 years in a country of over 200 million people. The broader incidence tells the same story from a different angle. 45 affected infants per 10,000 total births. In 1970, roughly 16,000 infants a year, falling to 21 per 10,000 by 1975, roughly 6,000, while over half gone in 5 years. Coverage climbed with it. The shot reached an estimated 80% of eligible Rh-negative women. In 1974, 81%. In 1975, 82% in 1976. Used correctly, before and after delivery, it is roughly 99% effective at preventing the sensitization that starts this entire disease. For our members on the Bloodline tier, the case file for this video breaks down the full surveillance data behind those numbers, year by year, plus the parts of the Sing trial that don't make it into most retellings of this story. A gene that had been quietly killing infants for as long as anyone had kept records was functionally solved within a decade of anyone understanding it, which raises a question this script has been holding off since the cold open. If carrying this gene was this dangerous for this long, why do 15% of Europeans still have it at all? And why does one specific population carry more of it than anywhere else on Earth? Here's the question the mechanism itself raises in its real population. Genetics, not a setup for mythology. Segment. Before 1968, an Rh negative woman with multiple Rh positive pregnancies was measurably more likely to lose children than one who wasn't. Chapter 5. The Bass Problem. That's negative selection by definition. Evolution working against a trait. Generation after generation, traits under that kind of pressure are supposed to become rare. Over enough centuries, they're supposed to disappear. They didn't. Roughly 15% of people of European descent carry the deletion. Today, one population sits far above that average. The Basque living across the Spain-France border, whose language isn't even related to any other in Europe. Basque Rh-negative frequency runs around 30% to 35%, the highest rate ever recorded on Earth, roughly double the European average in a trait natural selection should have been punishing for thousands of years. I want to be honest about where the science actually stands, because this is exactly the kind of question where other videos make up a confident answer that doesn't exist. Yet, there isn't one. There are real explanations on the table, and none of them is settled. The first is demographic, not adaptive, isolation, and genetic drift. The Basque are one of the most genetically distinct populations in Europe, likely descended from groups relatively isolated from the migrations that reshaped the rest of the continents. Gene pool, a trait common in an isolated founding population, can simply stay common. No selective advantage required, just isolation preserving what later arrivals diluted everywhere else. The second is adaptive, and it's more interesting. Heterozygote advantage. The hypothesis laid out by researcher Yaroslav Flegr and colleagues in PLOS, one in 2016, and again in the Journal of Evolutionary Biology in 2021 proposes that heterozygotes carrying one copy of each version may have better survival odds than people carrying two copies of either, which would explain how a trait, this costly stayed in balance instead of being bred out. But the evidence is limited, largely indirect, and in several studies, genotype wasn't directly measured, only inferred. A live, disputed hypothesis, not finding. Sequence work on the Basque Rh system, European Journal of Human Genetics, 2018, leaves both explanations standing. A 2026 preprint proposes a selection, model for the Iberian Peak, flagged as exactly that, a preprint on. Bioarxiv, not yet peer-reviewed. Now, let's deal directly with where some of you are already going. There are claims online that Rh-negative people descend from something other than ordinary human ancestry, extraterrestrials, the Anunnaki, Nephilim bloodlines, a royal or non-human line carrying special traits, higher intelligence, psychic, sensitivity, unusual eye color or body, temperature. Here's the actual genetics, laid out plainly. A deletion, a gene, simply going missing, is about the least exotic thing that can happen in a genome, and it happens constantly in every population on Earth. The geographic clustering, Europe and North Africa carrying more of it than elsewhere, is exactly what genetic drift produces everywhere in the genome in traits with no story attached at all. And the special traits claims fall apart under basic scrutiny, confirmation bias, from people who already know their own blood type, going looking for evidence. They're unusual in finding it because they're looking. An open question about why a frequency is high is not evidence for a specific exotic answer. It's an argument from ignorance, filling a real gap with the most dramatic story available instead of the honest one. We don't fully know yet, and ordinary genetics explains all of it without making anything up. I'm not saying this to mock anyone who's heard these claims before. Plenty of good, curious people land on this channel exactly because someone told them their blood said something special about who they are. So tell me, who told you that, and what exactly were you told? I want the real version in the comments because the true story about Rh-negative blood is stranger than any bloodline myth attached to it. And it isn't about where the blood came from at all. It's about where the medicine comes from, which is not a laboratory. Chapter 6, The Arm That Makes the Medicine. Here's a fact that surprises almost everyone, including people who have had this exact injection. The anti-D, antibody that stopped this disease, is not made in a lab. It's taken from human plasma donated again and again by living people, most of them Rh-negative men, deliberately and repeatedly immunized against the D antigen, specifically so their bodies keep producing the antibody the rest of us need. The cure for this disease is grown inside volunteers. It always has been. One of those volunteers deserves a name. James Harrison, an Australian donor, known as the man with the golden arm, gave plasma roughly every 2 weeks from 1954 until 2018 when he was forced to stop at 81, Australia's donor age limit, not his own choice. Over that span, he donated 1,173 times, credited with protecting an estimated 2.4 million babies, a scale that makes him one of the most quietly important blood donors in history. James Harrison died in his sleep on February 17, 2025, at 88. Now, here's the part that should unsettle you because it's happening. Right now, the United States has been in an active shortage of RHD immune globulin since December 2023, formally declared by the FDA, the ASHB, and the ABB in March 2024, caused by limited anti-D plasma supply, plus manufacturing problems at Kedrion Biopharma, one of only 4 companies licensed by the FDA to make this product at all for manufacturers for a shot every Rh-negative pregnant woman in America may need, and that's just the United States. A 2020 analysis in PLOS, one by Pegararo and colleagues, estimated that roughly 13 million doses of anti-D are needed globally each year for best prevention. Fewer than 4 million are actually given. Roughly half. The women who need this injection do not receive it. And among the countries analyzed, 100 have a supply gap above 80%. The disease the script described as functionally solved in 1968 is still, right now, killing and disabling infants across most of the planet for the single, most unglamorous reason possible. Not enough of the raw material exists. One, more pieces worth knowing because it's where the science keeps improving. Rather than giving every Rh-negative pregnant woman the shot, no matter what her fetus carries, newer programs test cell-free fetal DNA in the mother's blood to determine the fetus's Rhd status directly. So the scarce antibody only goes to women actually carrying an Rh-positive baby. Denmark made this national in 2010, reporting 99.9% sensitivity in its first 6 months. The Netherlands followed in 2011, Finland in 2014. If you or your mother ever received this exact injection and had no idea how close the system has come to running out of the plasma behind it, tell me in the comments. The disease that opened this script was never actually cured. It was pushed down hard by one idea in 1968, and it is still closing what your blood actually says, being held down today by nothing more solid than a supply chain. So here's what your own blood type actually tells you, stripped of everything this video just spent half an hour pulling apart. If you're Rh negative, it is a real medical fact with real consequences in one specific room, a delivery room where a doctor needs to know it to keep you and your child safe. That's the whole of it, not an identity, not an origin story written on your chromosomes, not a destiny, no matter what a corner of the internet keeps insisting. It is. Now widen the frame because that's the part worth sitting with after the video ends. For most of human history, before Frida, Gorman, and Pollock, before anyone had even connected the pattern, this exact mechanism was one of the most ordinary ways in the world to lose a child. Not rare, not exotic, ordinary, woven into the second and third pregnancies of countless families across every generation before 1968. Impossible to tell from the outside from simple bad luck or ordinary grief. Nobody could see it because it left no mark to point to. It looked exactly like tragedy is supposed to look. The cause set the whole time in the one place. Nobody thought to look for a killer, a mother's own blood, doing exactly what a healthy immune system is built to do, aimed at the wrong target. If knowing that changes how you think about a stillbirth in your own family tree, or a grandmother who was quietly told to stop having more children, subscribe, we go looking for the mechanism behind the grief nobody explained, and we don't stop until we can show our work. We dig into the mysteries that history tried to bury, and we don't make them up. Here's the last thing, and it's the part almost nobody tells you. The strangest fact here was never where Rh negative blood came from, or why the bass carries so much of it, or what a monkey has to do with any of it. The strangest fact is this: the deadliest thing this trait ever did to a family was fixed quietly with a needle inside a decade, and almost everyone watching this video has just heard for first time the full story of exactly how